Alright, so in this one we're going to talk about how to put genes inside eukaryotic organisms. So we want to alter the target gene and see the effect of it. So we have early blastocysts, and then we could take some of those pluripotent cells out, and you can culture them, and they'll stay pluripotent given the right conditions. You take out the differentiation factors so that they don't change and they remain as pluripotent cells or embryonic stem cells. And then you grow the embryonic cells, grow the embryonic stem cells in culture, and then you can alter it by introducing a target gene into the cells, and then you grow the colonies, and then you can test the colonies for your desired gene if it was incorporated or not, and how to make this how to do this genetic engineering, how to make this gene, we'll talk about it later, especially if you want to do a point mutation. There's a slide on that, how to make that. And so you PCR these colonies, and you use a probe to see if you have the gene you're looking for. And if you do, then you use that colony to grow some more, and then you take a couple of those cells and put them into a blastocyst. So you have a female mouse mate, then wait for 3 days, and then isolate the early embryo, and then inject these cells that have your gene of interest, and you let the embryo grow in a mouse, and it'll give birth to a chimeric mouse. And you choose a chimeric mouse, and the ones which have brown coat and white coat, and then you have these mice mate, and then you get a mouse that's all brown, then that's homozygous for the gene of interest, the gene you created. So this is going to be a knockout mouse because both copies of it have been replaced. And this is alright. So DNA can be introduced into embryonic stem cells in culture, and stably transformed embryonic stem cells can be isolated. So the DNA is being introduced into the embryonic stem cells, and you can isolate them using PCR and then a probe. These transformed cells can be injected into recipient blastocysts, where they are able to participate in normal development into the embryo. Put the cells into the blastocysts. Some of the progeny mice that develop after transfer of injected embryos to foster mothers contain cells derived as well as the normal cells. So basically, it's just saying what I said over here. And then inbreeding of these mice is going to give you a mouse like this that has 2 copies of the chromosome that you're interested in, to go to the chromosomes with your implanted gene. They're called knockout mice. And enriching for gene targeting events. So this one is better explained with the pictures. So you have your gene, and this gene is going to replace this part of the gene in where and this is the part, or the 3 billion base pairs there are. This has to find this exact spot and recombine with it. So obviously it's going to recombine in a lot of different places and also here. So you have 1 in 10,000 chance that it'll recombine right over here. So what you do is you use some controls so that you make sure that it recombines in the right spot. So the controls are neogene and the TK gene. So neomycin is to make sure that the gene itself is put in somewhere, not particularly at the homologous point. And the TK gene will make sure that it's placed in the right place. TK is thymidine kinase, which is going to alter gancillivir, and it's going to make gancillivir toxic. So any gene that has TK active is gonna, any cells that have TK active are gonna die. So if it does non-homologous recombination and TK is expressed, that cell is gonna die. So the only cells that are gonna live is the one that are homologously recombined. And if it's homologously recombined, the TK gene is disrupted, because over here we have the recombination at the ends, but over here you have recombination on the right side, not at the ends. So this TK gene will be disrupted due to it not being recombined at the ends. So cancellivir is not converted into toxin, and these cells won't die. So this is an example of this. So what's happening here is you have the wild type locus over here, and then you have your gene construct here. You have the TK here, thymidine kinase, the neomycin site over here, and you want to put this in here. And you do the same thing as back over here, and then it's going to recombine, and you're going to replace this gene with your gene, and you're going to select for the one that's been homologously recombined, and then you get this, and then the mutation you caused will give you this abnormality in limb development. That's the wild type. That's a mutant that you created with by changing the DNA sequence. And we said we're going to come back to how do you create a point mutation. To create a point mutation, you have your vector, the sequence from the, you can use a cDNA library. They're in plasmids, so that's a plasmid. And then the primers are normally 20 base pairs long, or the 20 base pairs you do a one base change one base. This is supposed to be A, but you put a C into it. You change the primer, so the primer will still bind sometimes, and then you let that you PCR that, you PCR this, and then you get this the first cycle. In the following cycles you get this and get some of this. This is the normal one, and this is the point mutation that you created. That's how you create a point mutation, and then you have a whole bunch of these plasmids, and then you can use this to do, so you genetically engineered a gene by creating a point mutation. So you created a point mutation, and that point mutation could be in this strand, and that's going to get inserted here, and then if it's inserted here, you create a point mutation in this animal. And then so over here, so the introduction of a point mutation in R722W. So this is R722W that's being targeted, and you put that in. with the use of the TK and the neomycin, and then doing that whole thing. And then you put it in, and then so that point mutation is going to cause the XPD, that skin disease that's due to sunlight damage. And then you see that the mice that have this mutation are gonna have premature aging. So you showed how to create a genetic disease